this model, it's blue or blue-green, right? So this is a triad. Both of these are triads. Sarcoplasmic reticulum, terminal cisternae, T-tubule, terminal cisternae. I'm going to take that off, okay? It releases its calcium. The sarcoplasmic reticulum releases its calcium, and the calcium comes down and it binds to these little green clusters called troponin. Troponin is associated with this little yellow line called tropomyosin. When calcium binds to troponin, troponin changes shape. It moves tropomyosin off of the sites. Each one of these little each one of these little red balls is an actin subunit. This whole thing is actin. Each little subunit has an active site on it. When the tropomyosin gets moved off of the active site of actin, the globular heads of myosin can bind to them. And then they, in turn, go through a power stroke and they push the actin molecules closer and closer and closer and closer and closer to one another. So the thin line gets smaller and smaller and smaller as the contraction occurs. When the calcium releases from calcitonin, the actin molecules slide past myosin until they're back at their resting length. And then we're ready to start all over again. As the actin molecules are moving closer and closer and closer to each other, you're generating force as the contraction continues.
Can you say that?
Sure. I want to introduce you two more players. This is the Z disc, where all the actin molecules are connected. This is the M line, where the myosin are connected. They look like a Z, so that's the Z disc. From one Z disc to another Z disc, all these players are considered one sarcomere. And the sarcomere shrinks as the muscle contracts. I'm sorry, what?
The Z-band and then what was the other thing?
Z-bands are connected to the actin. The M line
okay.
is connected to the myosin. So M for myosin.
Okay. And M is a
This
thick.
Z because it looks like a Z. And the myosin is the thick filament. So from here to here is the thick filament, and it's also the A band, darker band. And you'll notice as the contraction occurs, the A band doesn't get any bigger and it doesn't get any smaller, but the I band will get smaller and smaller and smaller and smaller. And there's some really neat. No, but whoever the model maker is pretty smart, because what we need to do next is get it so that the parts can move. That'd be neat.
You can work on it.
This costs about $800.
None of them have the elastic fiber.
I'm sorry, see?
None of them have the elastic fiber.
The elastic fiber is attached to, I believe it is, well, actually, I might have told them wrong. I think it's attached from here to here.
Yeah.
And as these contractions, these power strokes occur, and they move closer and closer and closer, these big pieces are actually turning. And as they turn, they coil up that elastic fiber called titin. So that when we're here, it's highly coiled, and it wants to go back to its normal unsprung size. And that's what determines the resting length. And it's a molecule called titin. You don't have to know about titin.
Can you say again when you said it releases calcium to go back to the electrolysis? Can you say that?
Sarcoplasmic reticulum, because of the action potential that speeds down the T-tubules, and the T-tubules are right next to the sarcoplasmic reticulum, the sarcoplasmic reticulum releases its calcium. The calcium diffuses down, binds to troponin. Troponin moves its shape, and when it does, it pulls on tropomyosin, moving tropomyosin off of the active site of actin. Once that active site is exposed, the myosin heads can bind to it. Then they go through the power stroke, and they push actin over its surface, or over its myosin heads.
Is it fair to say trypomyosin is like a blocker or like
It is. It's a
our
regulatory protein.
protector?
Yeah, it blocks the active site. If trypomyosin is covering the active site, actin and myosin can't bind, and you get no cross bridge formation. No cross bridge formation, no power stroke, no recovery stroke. You're just sitting there. That's why calcium is so important in this.
So are the active sites considered actin active sites or myosin active sites? Because that was confusing.
Yeah, that's a. I've seen it presented in
Either.
both ways.
Okay. Because I was like,
The important thing is where is the active site? It's on the actin.
yeah. Okay.
So you could say it's a myosin binding site on actin,
Yeah.
or you could say it's the active
Yeah.
site on actin. Either way, it's on actin.
And I have a question. Can you give me an example what you can be asking? Like, is the CETA, the M, like, what part is each one? Or you're gonna ask more like, we have to explain like how each one do and everything, or the lactose, or just only the identify.
I think I would probably just do identify.
Can you explain the relaxation?
Yeah, sure. Once the muscle is contracted, right? So we're looking at the contraction phase. That's where force gets generated. Now the calcium pops off of troponin, and it goes back into the sarcoplasmic reticulum. It's no longer there. Troponin changes its shape, allows tropomyosin to cover the active sites. Now actin and myosin can't bind, and so the muscle simply relaxes. It goes back to its original length. And its original length usually is a little bit so that the actin and myosins cover each other. Because if we go this far, if we go too much further, now they won't be able to bind. We won't get any contraction.
That whole thing's called a sarcomere.
Disease disc, sarcomere. And it is the structural and functional unit of a muscle cell.
You also have that picture in the extra
I don't
lab
remember if it's
supply.
in there yet. It may not be in there. This
The
is
new-
a new model that we got a couple years ago. And I have to be very careful with it because somebody had broken off one of these actins somewhere. Maybe somebody put it back on. It might have been this one.
It's the structural and functional unit of